Hello students, today in this video we will see 20 important MCQs for your university exams. They are very important for your university exams, and these important 20 MCQs were asked in previous year university exam papers of our university. So let me discuss from the first MCQ to the till the 20, and stay tuned till the 20th MCQ so that you will understand each and every MCQ clearly. And also please subscribe this channel to get more and more videos like this, and also share with your friends. Okay, fine. First MCQs will be which of the following, first MCQ will be which of the following cellular organelle is associated with signs and symptoms of gout. Here we have four options: Golgi bodies, lysosomes, microsomes and peroxisomes. The correct answer is peroxisomes, because peroxisomes having an enzyme called uric acid oxidase, which is absent in humans. So due to the absence of this enzyme, uric acid will be the end product and it elevates in the blood. It leads to hyperuricemia, but the same enzyme is absent in the reptiles, lizards and aquatic animals and all, so that they convert the uric acid into some other compounds and they can excrete. So peroxisome is the correct answer. The second MCQ will be which of the following clinical condition is associated with defective chloride channel. So chloride channel will be defective in following: multiple sclerosis and Parkinsonism, cystic fibrosis and Alzheimer's disease. The correct answer is C. See here, remember the correct answer. Always cystic fibrosis related questions are very important in case of MCQs. Chloride channel starts with C letter, and here the condition is also starts with C, cystic fibrosis. Cystic fibrosis is the correct answer. And now move on to the third MCQ. Coenzyme A is synthesized from. So the correct answer is the first one, A itself. So A, beta mercaptoethanolamine is a correct answer, and B, threonine is an amino acid, incorrect. Homocysteine is also an amino acid, is incorrect. Homoserine, all they are incorrect. Beta mercaptoethanolamine is a intermediate of sulfur containing amino acids cysteine and cysteine. So this intermediate gives us coenzyme A. And also pantothenic acid is also a source of coenzyme A. And now let me erase all this, and so that you can easily see the answer only. A. The fourth one, which is not involved in catabolism of heme. Cytochrome A involves, because it supplies heme to the catabolism of heme, and NADPH is also required. Incorrect. NADPH is an energy source for catabolism of heme, and again this cytochrome is also required, so incorrect. The one and only substance that we do not need for catabolism of heme is molecular oxygen. So molecular oxygen is a correct answer, and remaining all the cytochrome A, NADPH, cytochrome, all these are incorrect. So this is a correct answer. And here let me put a circle over here. May I erase it so that it will be clear for you all. See here, peroxisomes here. The fifth one, the primary immune response is due to. So the primary immune response is due to IgM, and IgG, this antibody crosses the placenta from mother to the baby. So placenta, which is incorrect, and IgA, which is present in tears, saliva and all, incorrect. And IgE will be involved in allergic reactions, so incorrect. The correct answer is IgM. As soon as the antigen enters, IgM releases into the blood. IgM releases and kills them off, so destructs them off. IgM destructs the antigens as soon as they enter. The sixth one is Benz John's proteinuria is seen in. So mostly Bence-Jones proteinuria is seen in multiple myeloma is the correct answer. Cirrhosis is incorrect. Chronic infection is incorrect. Heavy chain is also incorrect because multiple myeloma is a plasma cell cancer. Plasma cell cancer of bone. So in this condition, this is called multiple myeloma. In this condition, Bence-Jones proteins will be elevated. This is a marker. Bence-Jones proteins are markers for multiple myeloma, plasma cell cancer. So plasma cell cancer, for example, if an individual has plasma cell cancer, in his or her serum you can see Bence-Jones proteins. So the correct answer is multiple myeloma is the correct answer for 6th question. And now the 7th one of the following is not a high energy compound. See here, creatine phosphate, of course we all know this bond, a high energy bond is there. Creatine phosphate is a high energy compound. Just read carefully the question. One of the following is not high energy compound. This is high energy compound, creatine phosphate, incorrect. 1,3-bisphosphoglycerate is also high energy compound, incorrect. Arginine phosphate is also one of the most important high energy compound, not in humans, but it is present in reptiles and other animals. Arginine phosphate is also one of the high energy compound. Here the 4th one, D, is 2,3-bisphosphoglycerate is not a high energy compound. This is the correct answer. D is the correct answer for 7th one. So let me erase all these. Yeah. 8th, complex 4 is inhibited by the complex 4 of electron transport chain is inhibited by cyanide is a correct answer. BAL first complex will be inhibited. British anthracite, so anthracite, and 2,3-dinitrophenol is incorrect, and barbiturate also incorrect. This is also incorrect. So wholly cyanide is a correct answer. Okay, complex 4 is inhibited by the cyanide. So in cyanide poisoning patients, the cyanide in them will inhibit the complex 4 of their electron transport chain. Now move on to the 9th MCQ. Hexokinase enzyme belongs to the class of, see here, hydrolases, transferases, ligases and oxidoreductases. The correct answer is transferases. It transfers the phosphate group to glucose, so that glucose is converted to glucose 6-phosphate. So the correct answer is transferases. Km value in enzyme kinetics indicates the affinity of enzyme substrate, specificity of substrate, maximum velocity and saturating concentration of substrate. The correct answer is the D, saturating concentration of substrate. Km value of enzyme kinetics indicates the saturating concentration of substrate. We'll explain a separate video about the kinetics of enzymes. So now the 11th MCQ, the transport form of ammonia from muscle to liver is glutamine. So glutamine is an amino acid, is a storehouse of ammonia. After transamination and deamination, amino group will be loaded in the glutamine and transposed to the liver for the detoxification of ammonia into urea. So alanine is an amino acid which is incorrect, glutamic acid incorrect, alpha ketoglutarate is also incorrect. And the 12th one, methylation reaction are carried out with the help of, S-adenosyl methionine. We can also call it as SAM. So this SAM donates methyl group to the reactions wherever required, and methionine is not required. S-adenosyl homocysteine wrong, and cysteine is also incorrect. And the 13th one, at isoelectric pH the amino acids and proteins show, here you can see very important MCQ, they show maximum precipitability. So here C is the correct answer. Precipitability they show at a isoelectric pH. About this MCQ clearly I'll explain in a separate video. The 14th one is the substrate required to maintain the level of reduced glutathione in RBC is NADPH. NADPH is required for the maintaining the level of reduced glutathione in RBC. The maintenance of reduced glutathione in RBC helps the RBC membrane integrity. So NADPH is required. This NADPH, RBC gets this NADPH from HMP shunt. Remember this one, very important. Okay, the 15th one, the enzyme that converts ATP to CAMP is Adenylate cyclase or adenyl cyclase is the correct answer. Phosphokinase, phosphorylase, phosphodiesterase, all these are incorrect, randomly given. The 16th one, NADPH is produced by the glycolysis, no. Citric acid cycle, no. NADH plus H plus will be produced in these two. Glycogenesis, no. Synthesis, no. HMP shunt is the correct answer. HMP shunt is a source for NADPH, and that NADPH is in turn required for biosynthetic reactions of metabolism. And the 17th one, bisphosphoglycerate shunt is seen in bisphospho 2,3-BPG, bisphosphoglycerate 2,3-BPG shunt is seen in Rapaport-Lubring cycle. So the pathway which produces 2,3-BPG is Rapaport-Lubring cycle. 2,3-BPG is required for unloading the oxygen from RBC to the tissues. RBC to the tissues. Remember, 2,3-BPG is very important compound produced by Rapaport-Lubring cycle. And the 18th one, the major glucose transporter in skeletal muscle and adipose tissue is A, GLUT1, GLUT2, GLUT3, GLUT4. The correct answer is GLUT4. GLUT4 requires insulin to be opened. Okay, insulin activates the GLUT4, so that GLUT4 channel opens and glucose enters inside. So GLUT4 is the correct answer. GLUT1, GLUT2, GLUT3, they are not. GLUT2 is present in intestine. And also we have to know GLUT1 and about all these GLUT means glucose transporters. About all these we'll discuss in a separate video. 19th, glycosaminoglycans present in a cartilage. Glycosaminoglycans are a classes of mucopolysaccharides, type of mucopolysaccharide. In a cartilage, glycosaminoglycan is present among these four. Like one of the glycosaminoglycan among these four is present in a cartilage, which is chondroitin sulfate. Remember, in this way, cartilage starts with C letter, and the glycosaminoglycan is also starts with chondroitin sulfate C. So C stands for cartilage. Whatever glycosaminoglycan is starts with the C, that is for cartilage. And last one, 20th MCQ, the identification test for galactose is second epimer of D-glucose. See, isomerization reactions of carbohydrate. The correct answer is D-mannose. You can go through the reaction later. D-mannose is the correct answer, and D-galactose, fructose, sucrose is, all these are incorrect. So this is all about the 20 MCQs answers, discussion and everything. You can just see and correct and just remember, just keep it in mind for your exams. Yeah, you can just pause the video and see, but don't forget to subscribe and share with your friends. Thank you.